So today I'm going to talk about, this is a project that's been ongoing in my lab for quite some time now, and it's using what we find in some women who have present with autoantibodies to proteins that are present in the developing brain, and that we have turned this sort of into the ability to detect risk for having a child with autism. So, I hate when, here we go. So we all know what autism is. Clearly, we've had many talks on that, and many of the people here are experts. But I think this is sort of a new way of looking at autism, and it's not new anymore because we've been doing it for quite a while now. But it's sort of the maternal gestational inflammatory environment has been shown to alter neurodevelopment, and I think when we look at our animal models of those, I think we have to be very cautious in saying, oh, it's autism or it's schizophrenia. I think right now we're at the level of saying, yes, we can change how the brain develops with maternal immune activation. And I think, which is certainly exciting and of interest to many of us. Just a quick brief background, autoantibodies are antibodies that recognize self-proteins. They are more prevalent in women, unfortunately. We seem to carry a higher risk for those. But we also know that there's precedent for maternal autoantibodies being present during pregnancy, crossing the placenta as they do during beginning about 100 day 110 of gestation. And unfortunately, when they go across the placenta with the healthy antibodies, they can cause fetal effects. And so I think probably historically one of the best known of these is SLE, where antibodies that cross the placenta during gestation affect how the heart develops, and they can end up with a congenital heart block. So just to give you a quick background on who we are and what we do, we're looking at the roles of environmental factors, genetics, and the immune system as they relate to autism susceptibility. We do this through our large population studies. I'm lucky to work with several epidemiologists and our animal models, and we address how the environment can trigger immune dysregulation and affect brain development. And we do, some of our work is in the kids, and some of our work is during the gestational period. We also are looking for how biologic markers, such as those related to the immune system, can play a role in ASD, either at the diagnostic level or trying to help understand mechanisms, as Matt was already just talking about. To understand the role of the environment in immune dysregulation, I think, is a big part of our program. We had the Center for Children's Environmental Health, and that was a focus of that program. But the gestational immune environment has turned out to be, I think, one of the most fascinating because of all the sort of the ballet that goes on during neurodevelopment, and that sort of back and forth between the immune system and the developing nervous system, and those are highly integrated, and some of the same molecules are performing similar tasks in both those compartments. So we look at how changes in immune signaling molecules such as cytokines and chemokines affect neurodevelopment, and that's a whole nother prong of our research. But today I'm going to talk about the maternal autoantibodies to the proteins in the developing brain and how their potential role in autism. So just to give you a quick overview of what this model is, and as I said, some women develop antibodies to their own proteins. These are what we call autoantibodies. And along with the healthy IgG that crosses, it's only IgG that crosses the placenta, these antibodies unfortunately go along for the ride, and they can recognize and bind to their targets in the developing brain. That these antibodies get inside, can get inside, this is a radioglial stem cell, inside these cells, and it changes the way they migrate, the way they replicate, and changes how they develop. And you can see this is from a mouse that was a control animal, and this is from one of our MAR exposed mice, and you can see the dendritic spines look completely different, or they're almost absent. This is about 20 to 26% of cases, and I say there's a range there because it completely depends upon the population that we're looking at. Some of it's geographically different populations, some of it is whether it's during pregnancy in a high-risk population or in the postnatal population. So it is a distinct behavioral subtype, at least to date. These are the kids that are usually more severe. The kids with more severe behaviors, they have pronounced stereotypic behaviors, and significantly larger brain volume in boys. So the population that I'm going to talk about, just the clinical study that we just completed and just was accepted for publication, is a study that we did in the CHARGE study. This is run by Irva Hertz-Picciotto here at UC Davis. And this is really, it's a postnatal population. So the maternal samples were taken at the time of the assessment of the child at the UC Davis Mind Institute. The kids are actually, it's 2 to 5, I should have put in there. This is a retrospective study population, meaning the child, we're taking the maternal sample postnatally, but we did see, I'll show you the data from that study, and we do have prospective studies that are currently in data analysis. So we've done a huge number of samples during gestation that we're just finalizing the data analysis on that. But for the study that we just completed, and I think this study, the goal of the study, we've published on these autoantibodies quite some time ago, but the current study really was, we created an ELISA test using the proteins that we discovered in 2013 that we published on then, but we also used machine learning to identify which patterns were highly associated with autism. And when I say highly associated, I mean, we have 0 TDs in this population. So all the cases that we see where the mothers have these autoantibodies are ASD cases. And so we made a very stringent assay. The cutoff was very stringent for this. So we intentionally went after that 100% specificity. We know that may not last for all of these, but at least that's where we stand right now. And that led us to 8 major, what I call backbone patterns, because rarely do we see just 2 of these alone. Sometimes we'll have 2 patterns combined, like a common one is CRIMP1, CRIMP2, and NSE and STIP1. So these proteins are, I didn't really go deeply into what the proteins are, but each of these has a role in normal neurodevelopment. And so we know that if we impact these, that we see an outcome of autism. The odds ratio associated with this is, 31 is the highest I've seen, in anything associated with autism, down to 10. But this means, some of this is because of the numbers that we did. There's about 850 samples that were in this study. I think you can see there's 450 ASDs that we had in this population, and about 360 TDs. But this is where this is right now. And I think, a big question for us is always, are they pathogenic? The data I was just showing you lends support to them being an excellent biomarker of risk of a child developing autism. But we really need to know if they're pathogenic, because that tells us that if we determine they're pathogenic or they have a pathologic significance, we can then look at, if we block them, do we actually effectively develop a treatment? And so we have established that the maternal autoantibodies induce autism in animal model using both passive rodent and monkey models. We've completed those studies. Those are all published. Do they localize to the brain? We've shown that as well. We show these antibodies do cross placenta and get into the fetal brain. And then the important thing is create clinically relevant animal models wherein the dams produce the autoantibodies themselves. We aren't passively transferring human antibodies in. So we call this an endogenous model that mimics the reactivity found in humans. And this is a really important point for this. So I will talk about that more in a minute. So working with my colleague, Melissa Bauman at UC Davis, we used the most characteristic antibody pattern that we saw that was the highest significance was associated with lactate dehydrogenase, CRIMP1, and STIP1. And so we saw that there was increased stereotypic behavior associated with those antibodies. We did a preclinical study where we passively transferred 6 times beginning late in the first trimester. So just as the antibodies are getting ready to start being able to cross the placenta in rhesus macaques. And we found that really interesting. There were a couple interesting things. One is the monkeys would go up to their conspecifics and would approach, but whatever they were projecting in their behavior was seen as odd by the control animal. So you ended up with what we call affiliative behaviors lower in this population, going the wrong direction, versus the more times you come up and you sort of get a reciprocal interactions between the two animals. So I think this was a really interesting study. We are now creating the endogenous monkey model. So we're moving past all of the passive transfer, but it's cheaper and a great way to start. We also saw in these animals, I think this is one of the most exciting findings that we saw, was that they had enlarged brains throughout development. And this is sort of a graphic depiction of that. And one of the things that makes this so interesting is this is what we see in the kids whose mothers have these autoantibodies. And on the left is a population of children with autism whose mothers are negative. And you see quite a range of brain size. And this is work we did with Christine Nordahl. In the typically developing population, you see also somewhat of a range. And these are statistically different from each other. Well, in the middle here, in this very tight group, are the MAR ASD, kids that came from mothers who had these autoantibodies. And they differ statistically significantly from either population. So this sort of sets them apart. We've seen this in our monkey model. We see it in our mouse model. We see it in our rat model. Everything we've done, this has been the finding that has come through all of those studies. So as I said, the better model for us is to use the clinically identified epitopes, meaning the parts of the protein that the human antibodies bind to. Because when we replicate a model, when we create this model, we want it to replicate what we see in the humans. And if I just took the whole protein, threw it in there, the animals are not going to make it to the same part of the protein, and this would not be a valid model. So we began with the lactate dehydrogenase STIP1 and CRIMP1, and the antibodies now are present throughout gestation and lactation. So these antibodies are made by the dams before we ever breed them. As I said, we epitope mapped all 8 of these proteins. We began with the LDHA, MB, CRIMP1, STIP1. We waited until the dams had a really good titer to these proteins. The controls had saline plus adjuvant. But this is our MAR population. And then we began immunizing well before breeding. And this is an important distinction between the maternal immune activation model, in which case we're giving them poly-IC during gestation around day 12. And these animals, so there's no inflammation in the dams. We've tested that. They have no elevated cytokine levels, nothing at the time that they are bred. And that is a really also important part, because we don't want to be mixing up our models. And we really aren't looking at when they're initiated. We're looking at if they're present, what is the outcome? One of the things that we see, as I said, is that we see this distinct difference in how some of the neurons develop. And it's not throughout the brain. There are some differences, but this was from a passive transfer model. But we do see similar findings in our active immunization model, where we see that there's this lack of and the dendrites, the lack of spines density is profound in these animals. And then just one of the overwhelming findings was the lack of the decreased sociability. So I'll play a movie here and you'll see the control animal. We've got a stimulus mouse in there, and these are juveniles, and we have the first movie is of the saline adjuvant treated animal. So you can see there's these are mice, so they're not terribly, you'll see the rat video in a minute, they're completely different, but they've got some social behaviors: push, crawl, front approach, following, and lots of sniffing. And so you can see there's a lot of interaction between the two. Actually, our stimulus mouse is grooming there, but you'll see in a second this will change when we have the MAR ASD animal. And I don't even have to have the arrow on there, you can tell which one it is, because the stereotypic, they're what we call repetitive behavior, not stereotypic, but the repetitive behavior, self-grooming, is so profound in this animal. That's all it wants to do. It walks away from the stimulus mouse to be by itself and just spends its time self-grooming, because this is a stressful situation for these animals being put in this cage. So I think this was one of our and I didn't have time to put all of graphs in here, but so we then created a rat model. And one of the reasons we went to a rat model is because their social behaviors, first of all, they have some prefrontal cortex, but their social behaviors are so complex, it gives us a much better way of really doing a fine dissection of the behaviors that are impacted. The other thing I want to mention before I play the movie is that rats don't do stereotypic behavior like mice, the self-grooming thing, unless they are these rats. And our staff that was doing the behavior analysis thought we had gotten a bad batch of rats. They had no idea, what they were testing. So these are two control animals, one adjuvant saline and a stimulus animal. And you can see they have very robust play behavior and lots of mounting behavior, and they're very active. So this is an important aspect of these animals. And then we have, you'll see the Mara ASD animal, and you can see if you can guess which one it is, but interesting, similar to the mouse, it's just not interested in playing. And the stimulus rat tries for a while, and this is a difference between the mice and the rats, is that the stimulus rat, which is the one on the right here, gives up. And the Mara ASD rat is just licking the bottom of the cage. So we saw deficits in social, we saw reduced social interactions, reduced play, increased repetitive behaviors, which we don't normally see in a rat, and reduced ultrasonic vocalizations when they were young. So this really was profound in the juvenile because that's when they play the most, but it did hold up throughout these animals. So, this is where we are at a point where we're having to make that that transition from academia to industry. And I think, where do we fit in here? And as we know, that the screening and identification of neurodevelopmental disorders happens very early, but really it starts in the well baby visits, and you start seeing, okay, were they failing to meet milestones? And the beauty of this, that we test the mom, not the child, for one thing, and we can do it at birth. If we thought someone was at high risk of having a child with autism, we could do it prior to conception, because it is not a fetal-based test. And so just to give an idea of how having this potentially could change getting these kids into earlier treatment would, especially, this is where we're thinking, and in terms, now I'm wearing sort of the company hat, but that MAR test, if we see anything on the MCHAT or any of the early screeners suggesting, and we could even pair it with some of the other technologies that have been shown today, you could test and you would know and get those kids into earlier intervention. Unfortunately, it's we're only talking 20, 25%, 26% of kids, but that's still to us, in this space is a large percentage of the population. And then if you saw anything, the earliest you could do, you could get these kids in the earliest treatment. And so, failing to meet eye gaze, any of those early signs. So just a quick conclusions. I think we've identified what it doesn't seem very new to us now, but new paradigm for autism etiology. We've created the ELISA, the testing parameters are completed. We've got everything in production. We've done over 850 samples that we did with that to create that test and validate that test. And I'm not talking clinical validation, I'm talking assay validation. This would be and so far we have 100% specificity. So I think I expect that to change. We never say we have 100% of anything, but I think we're really lucky in that we've created a very tight assay for this. And I think we're thinking CLIA for this. So that I just wanted to give you that as a perspective, given what the topic was for this, that we're thinking in terms of potentially using this. But I also want to thank, I have an amazing team of people, and there's just some of them shown here. We were lucky to be supported by so many people, especially the families that have donated their time and their samples to us. And I'm happy to take any questions.
So we've got a couple of questions for you. You touched on this a little bit, but is that sort of, I guess, your corporate goals to end up having the MAR test at birth, like a PKU test?
I think that would be fantastic. I think there's a lot of data to be collected before that could happen. However, I think our prenatal studies, I think the beauty of this test is we don't have to do it as a prenatal screen during pregnancy, which has its own, I mean, we would prefer not to do that, and we are not going to do it that way, but we have the preconception testing we could do, and we have the early postnatal testing that we can do. So I think we're sort of in that place that if we wanted to look at very early at birth as a newborn screen, absolutely we can do that. I think especially for women that are high risk, and there's a number of factors that make you high risk. Metabolic syndrome during pregnancy is one of them. So we know there's a link between these autoantibodies and those syndromes. So I think, our hope is that we are able to do that. I think the most important thing is getting kids what they need as quickly as possible.
Great. And if you can put on your prognostic hat for the future, so what sort of treatments do you think may be on the horizon with a MAR test? What do you think could be tied to it from a biological treatment perspective?
From a biological treatment, we've got a couple of things in the works. So there's a couple of possibilities, and I think, some of which hinge on blocking the antibodies from getting across the placenta. That is done for some other diseases of the newborn. So I think, there is precedent for that. In the postnatal, if you already have a child, and we've done some downstream RNA-seq work on the brains of these rats, and we have a number of targets that are potential for treatment, because we know what the antibodies target. For us, an important aspect was what other pathways do they change? Because there are downstream effects of these, and we've found a number of them, some of which, so there's another paper that's in the works, is the rat model paper has some of this in it, because we've done imaging and we're seeing MRS changes. We're seeing pathways, things in the brain that are changing, analytes changing. So I think that's my hope, is that we're going to actually hit it from both sides. Prevention, if we get it prior to conception, and postnatal treatments based on some of the molecular targets that we're looking at.
Great. Another question, do you think the antibodies are a byproduct of pregnancy, or do you think it may be possible that some of these MAR antibodies may be present in these women even pre-pregnancy?
I don't think they're necessarily pregnancy generated from some samples that we've ended up testing in the lab at the research level from individuals who've never been pregnant that are positive, but they have other things like inflammatory bowel disease. So there are other, sort of what I call immune-mediated disorder indications. We don't always use the word autoimmunity for those, but immune, inflammatory or immune-mediated disorders. So I think we don't have enough for me to make a concrete statement. And it's the $1 million, $10 million question, is what causes them to be made. But I think there's some interest in looking at that further.
Great. To that point, in terms of possible what may be causing these things, have you guys looked at microbiome profiles and seeing if there's anything there that may be a signal that may predispose someone to the antibodies, or any work related to the microbiome with these antibodies?
No, I think that is a really interesting link that we, at the time we did our study, we didn't have the means to do that. We didn't have the materials, for the women that I just finished the study on. For the MARBLE study, we have microbiome on the infant that we could look at. And I don't know that they've done the mothers, but I know we have meconium, we have, materials that should reflect what is going on in the mom, so that we can link that. So the MARBLE study is the mothers at risk, so that have already had a child with autism. And we've got 2 studies that we're finishing up, and that's part of our IDDRC renewal. So that project will be continued through that.